Hello, everyone. Welcome to our monthly Science of Alt Protein seminars. My name is Amy Huang, and I'm the Senior Associate Director of Scientific Ecosystems here at the Good Food Institute. For those of you who are here for the very first time, I'm so grateful to welcome you to our community. The Good Food Institute is a nonprofit think tank that is 100% driven by philanthropy, and our singular goal is to build a world where alternative proteins are no longer alternative. And we see all of you as really central to that mission. So if you are looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, check out our open access resources at gfi.org/science. We have a whole lot of curated resources that exist to help scientists trying to find their way through the alt protein space. With that, I am so excited to introduce today's speaker to you all. Dr. Cathy Walsh is a senior scientist and program manager for the Marine Immunology Program at Mote Marine Laboratory in Sarasota, Florida. She obtained her PhD from Clemson University and has been a research scientist at Mote Marine for 34 years. Her research focuses on basic and applied research on the immune systems of marine vertebrates, including cartilaginous fishes, Florida manatees, and sea turtles, which all sound so delightful. Basic immunological research with marine species explores how immune systems from different species function and potentially lead to novel insights into the evolution of the immune system. And Dr. Walsh's current research projects explore how many environmental stressors, such as red tide toxins or brevetoxins, can really impact the immune health of marine species. Her more recent research projects focus on the development of cell lines from marine finfish and inverts, which she'll be discussing at length in this seminar. So Dr. Walsh, I'm so grateful that you're joining us today. Super excited to learn from you, and I will turn the floor over to you.
All right. Well, thank you very much, Amy. Thank you, everybody, for joining us today. I'm going to share some of the more recent research that I've been doing over the last 3 or 4 years about cell cultures with different marine species that have relevance for cellular agriculture or cellular seafood. And again, I'm based at Mote Marine Laboratory in Sarasota, Florida. The two species that I'm going to be discussing today are the Pacific white shrimp, Litopenaeus vannamei, and a marine invertebrate, and a marine teleost fish, the red drum, Sciaenops ocellatus. I know that all of you are working in this area, so I don't have to spend too much time on the importance of cellular, our overall objectives of cellular agriculture, but I did want to share, the importance of shrimp for seafood, because Litopenaeus vannamei is the dominant crustacean species that's grown in aquaculture on a global scale. So it's a really important species and a, ideal species for, wanting to develop for different cellular agricultural processes. And the production of the species is one of the fastest growing food crops globally as well. And of course, we all know that shrimp are very tasty, so they make a really good candidate for this type of research. I wanted to share, today I'm going to share like the research that I've been doing with the shrimp for the last few years. And basically, our approach was to use the fertilized embryos from the shrimp. So we take the eggs that are fertilized and we take the embryos and we're isolating out the cells at the blastomere stage. Again, our objective was to develop a cell line from those embryonic stem cells that are in the eggs at that stage, with the idea being that those cells are highly proliferative and they have the potential to develop into different cell types. So our hypothesis was that would be a good source to develop a cell line from. This picture here shows the embryos on the right, and those are they're about 300 microns, a little over 300 microns in diameter. So they're kind of small. I think some people might be used to working with larger eggs, but they're small, and this is what we're using. And again, the shrimp are in the same tank, and they're fertilizing. So the batches of eggs that we get are from multiple animals at the same time. So our research goal, we collected our fertilized eggs. We get them from a shrimp farm that's not too far from here. And then our goal was to develop the cells for a cell line. And again, our hypothesis was that by using these cells at the blastomere stage, they would be highly proliferative and have the potential to turn into a variety of different cell types. And we wanted to develop aseptic protocols to isolate the cells from the fertilized eggs at this stage, and then also develop different media formulations for successful growth of the cells and protocols and things like that once we got the cells out of the embryos, out of the fertilized eggs. But honestly, we did have a lot of challenges with that, and I'm going to share you the details of some of those challenges. But I also had this publication that came out in early 2025. So most of the information that I'm going to share with you today in this talk, you can find out more details if you want to take a look at this publication as well in vitro cellular and developmental biology. So before we start on the actual protocols, I wanted to mention that the osmolarity of the shrimp system is higher than it is for, teleost fish or mammalian species that you're working with, that you might be working with. They have about 700 milliosmolar in their physiological fluids. So we have to adjust our reagents to be to that osmolarity so that the cells, are in the proper osmolarity for survival. So we make our reagents, basically our PBS is a regular PBS that we just supplement with sodium chloride to increase osmolarity to the proper level. And then we also have another reagent. I'm going to, defining these here because I'm going to use these terms in a little bit, and I want to make sure everybody understands what I'm talking about. But we have a shrimp PBS plus, which we actually supplement with penstrep and amphotericin B to help control microbial growth during the cell isolation. Okay, I hope everyone can see this because it's a busy slide and it's detailed. But then, again, this is in that paper too. But if you look in the upper left-hand corner, you can see a beaker that is full of eggs. So these are the eggs that we collect from the shrimp. We can get quite a bit. And then there's the picture of the eggs under the microscope at that stage. And then in the middle there, you can see a beaker pouring the eggs through a sieve. So basically, we take those eggs and we concentrate them in the sieve. The sieve is really just a PVC piping with a nylon mesh super glued to the bottom of it. The nylon mesh is 100 microns, for the openings. And we sterilize this in like a 70% ethanol solution before we pour the eggs into that. But that's how we concentrate the eggs. And then over to the right, you see a series of Petri dishes. And in these Petri dishes, we put our shrimp reagents. And the first one you have, shrimp PBS, we do a series of washes, like we swirl the sieve into the Petri dish to, separate the eggs and, swirl them around to make sure that, they're getting washed on all the surfaces and they're separated. And then the final wash is the shrimp PBS plus that has the penstrep and amphotericin B to, decontaminate the egg surfaces. I actually forgot to tell you, the eggs actually, we are getting these from a shrimp farm that uses these eggs for commercial purposes. And so they have very sterile procedures, and they actually bring the eggs to us treated with different decontamination solutions so that they're, shouldn't have any contamination on them from their procedures. But basically then the last step here is the penstrep and amphotericin B mixture. And then if you can see these gloved hands and that PVC pipe with a sieve on the bottom, I don't know if you can tell, but there's that sterile borosilicate vial is what we use to crush the eggs. So we take that vial and we literally press the eggs down into the Petri dish, through the sieve, and crush them open to release the cells. So the cell debris then stays above on the sieve, and the cells are small enough that they go through into the Petri dish below. We collect that into fluid below. And then the next step, you can take it, take that and put it this solution down below in the Petri dish and that contains the cells, and put that through a filtration. It's just a regular filtration used for cell culture. That's a 40 micron size in there. They're sterile, and you put it on top of the like a 50 mil centrifuge tubes, and you filter it through there so that the egg debris remains on top, and only the cells then go through. So any egg debris that might have passed through then is caught by the 40 micron, so you're just left with cells. And this is a picture that we took under the microscope of our fertilized shrimp embryos before we isolate the cells. And this is the cells once we isolate them and under a hemocytometer. So this is what we're going to be counting to, put the cells in culture. And you can see there's a lot of really small cells, but then there's these occasional larger cells that look like aggregates of the smaller cells. And then once you put them into cell culture, you can see that there is a lot of smaller individual cells. And there's also a number of these larger cells that appear to be the aggregates of the smaller cells. So that's something I wasn't used to looking at with many of the other cell cultures that I have worked with over the years, because I have worked with cell cultures from many different species. Basically, for starting our cell cultures, we start them in 24 well plates. We seed them at about 50,000 cells per well. And as they expand, we move them to 6 well plates first and then onto 10 mil flasks. But this way we can test different media simultaneously and things like that, and select for ones that are growing well. We did test a number of different media, but for the most part, we focused on using Agrace's insect media. That is basically, it's, from Gibco, and it has supplementation in it too. And it's 700 milliosmolar that we adjusted again with sodium chloride. We add some growth factors such as leukemia inhibitory factor, again, antibiotics and antimycotics. And we also were using a filter fetal bovine serum at a high concentration for these studies. Now I use a lower concentration, but at the time we were using a 20% fetal bovine serum. And this is, of course, all filter sterilized before using it in the cell culture. We tried a bunch of, I know this is a busy slide, but we tried different supplementations that included different growth factors, shrimp mussel extract, and some other amino acids and different levels of fetal bovine serum. But that serum, that media formulation that I showed you before was the one that we used for most of our experiments. All right, now I'm going to show you what the shrimp cells look like over time. So to be honest, this was my, again, I had mentioned that I had done research with a lot of cells from different species, all different kinds of marine species, but not really with invertebrates. So this was my first work with invertebrates. And so I wasn't totally familiar with what I really should be observing under the microscope. But this is what I did observe. So on day 0, this is basically that same slide where we had, that I showed you before. We had a lot of small cells and then the larger aggregates of the bigger cells. By day 1, you can see that we're getting more of the aggregates of the small cells forming. And then on day 6, you can see that all the cells are now looking like they're aggregates of small cells. By day 7, I'm sorry, because I don't have a pointer, but up towards the top, you can see that there is a cell with a clear cytoplasm. You have most of the cells are the getting to be more uniform in size, but then you see the one with the clear cytoplasm and the dark nucleus, and that's those are cells that are become more predominant at days 7 through 10. This is day 8 that doesn't show that, but it shows some of the aggregates again. But here on day 12, you can see there's more of those cells where you have a clear cytoplasm and a large, big nucleus. So I'm seeing a lot of different cells in here. And so I wasn't really, understanding, focus on what I should be clear about what I should be focusing on. Again, I hadn't really worked with the shrimp before, but I was happy that something was growing in the cultures. And this is day 14. You still have those bigger cells with the dark nucleus growing and the smaller cells there. In day 20, and day 38, by day 130, you're not seeing those big black cells anymore, the big dark cells, but you do see the cells are more uniform and they're starting to form these loose clusters. So these are all non-adherent. And so I thought I was working with like a suspension cell culture. And then that's what persists is these clusters, those grape-like clusters of cells over time, because I kept this culture growing for quite a while, and they're more uniform in size now, and then up to 207 days you can see they're all more similar now. But and so I started to be very concerned that I wasn't growing what I thought I was growing. And in my defense, it was because, the cells, there were so many different morphologies that were present in the cells, it was hard to understand what really was going on. Because, you have the grape-like clusters that were there. In the center of this slide, you can see that there's a large cell in the middle that has a large nucleus and a really thin cytoplasm. And then you have those dark nucleus cells with abundant cytoplasm. And then you have these larger clusters. But I did want to point out, so there's a lot of different things going on as far as cell-wise in here. But then if you look underneath, you're starting to see, you can see it better in this picture. You see some cells that are growing underneath. They're darker and they're round, but they seem to be growing underneath. Now they seem like it look like they might be adherent, but they're actually not. They're just really loosely adherent. And I bring this up because, well, because I was really focusing on the suspension cells, and in retrospect, I wish I would have given more considerations to those ones underneath after the fact. Okay, and then around day 8, you're already seeing you see these dark cells like almost every time starting to form around 8 days. And the aggregates. So there was a lot of things that were happening in these cell cultures. So, it was a challenge to figure out what really to focus on. And again, you start some of these other cultures, you can see the dark cells underneath. I did try a bunch of different types of plate coatings, but they weren't really adherent. So the first picture here shows non-adherent cells, and B shows ones that I adhere to laminin treated, and they did loosely adhere. And C is polyallyl lysine. A lot of things did loosely adhere, but they weren't like you certainly didn't have to use trypsin or anything to remove them. You could just like pipette them gently and they would come off. We also tried a chitin, and actually if you look at the last 2 pictures here, E and F, those are both on untreated plates. So it really didn't improve the adherence of the cells to use the different kinds of plate coatings. They adhered to the untreated plate just as well, but they were always loosely adherent. They weren't like a strong adherent cell type that you could feel confident separating from the rest. All right. So again, midway through all these cultures, I started to suspect that maybe I wasn't really working with shrimp cells that I thought I was. Even though I got them straight from the fertilized embryo from a shrimp, it seems like something else was growing instead. So one of the first attempts that I did was 18S community analysis to try to determine the relative abundance of the different taxa in our shrimp cell cultures over time. And we did this, the analysis was done by Mr. DNA. We sent the DNA to them for their analysis. And what you can see here is on day 1, if you can see the legend over there too, those are all shrimp there through this 18S community analysis. Day 1 and day 3 are coming up 100% shrimp. On day 5, you're starting to see fungi that are starting to creep into the culture. On day 15, you're having more. And then on the longer term cultures, day 49 and 157, they're 100% thruster chytrids. So if you're like me, I had no idea what a thruster chytrid was. And so that was something I had to do a lot of research on, but it turns out in the later cultures, that's 100% what was in my cell cultures. It's very disappointing. Okay, so we also, the 18S community analysis was very helpful for us understanding what was in our cultures, but it takes about a month to isolate DNA, send it off, and have the data come back. So it's really not practical for monitoring cell cultures over time. So we came up with a different approach that we developed our own primers for because CO1 barcoding is a really common way to identify species in cell culture. So we developed primers for the cytochrome oxidase gene for shrimp, and also for a thruster chytrid species that showed up in our community, our 18S community analysis that we conducted, so that we could use this as a quicker way to identify species in our cultures. And so just to show you, on day 0, we have in lane 2 and lane 7 is the same day 0 sample. It was positive using our shrimp CO1 primers, and it was negative for the thruster chytrid primers. We did a replicate culture at the same time, and again, it was positive for the CO1 and negative for the thruster chytrid. On day 1, we did see the same thing, but on a later day culture, this is a big jump in days, but on day 56, you can see that there is nothing in this showing up with the shrimp primers, and we have a definitive band with our thruster chytrid primers. So these primers are something that we've developed that are useful for identifying shrimp in our cultures. And again, the sequences for these primers are in that paper if you wanted to look them up and see which primers we used. This slide here is a comparison of the results from the 18S community analysis and our CO1 barcoding studies. And the days in culture are on that left column. On day 1, we had, with the community analysis, it was 99% shrimp. It was positive with our barcoding for shrimp and negative for the thrustochitrid. On day 5, you're actually, with the community analysis, you're seeing that there is a decrease in the amount of shrimp, and you actually are starting to see an increase in the thrustochitrids at that time. But with our barcoding, it shows a positive for shrimp and negative for thrustochitrid. And then on day 15, you're seeing a positive for both shrimp and thrustochitrids, and they are mixed population through community analysis. And at day 18, it's 99% thrustochitrid, and it's negative for shrimp and positive for the thrustochitrids. So these results correlate very well with what we're seeing with our shrimp primers. Okay, so I mentioned this a couple slides ago, but if you're like me, I was not familiar with thrustochitrids. I'm familiar with other kinds of contamination that can occur in cell cultures. Bacteria is probably one of the more common one. Definitely yeast and some other kinds of things. But thrustochitrids is not one that I had ever encountered before, and I had to do a lot of research on what they were because I wasn't familiar with them, and I actually only ever even thought about those being in my culture because of the work that we were doing with the community analysis and the the CO1 barcoding. But what I had found, I scoured the literature for all these information about them in cell cultures, and what I did find is that marine invertebrate cell lines are actually frequently complicated by the appearance of these organisms. So, I was not alone in that. There's several different reports of that happening in different marine invertebrate cell culture attempts. But they are a very common group of unicellular marine eukaryotic protists. And they have within the class Labyrinthomycetes, and they have a number of different genera. So they're diverse, and it's hard to identify individual genera and stuff like that in your cultures. They have a very complicated life cycle, and they have multiple stages and multiple morphology. So it makes it hard to understand where you are in their cycle. And also I think it complicates being able to eradicate them as well. They're known to be saprivores. They're common in water columns, sediments, marine organisms. They feed on decaying organic material, so they play important ecological roles to help break down some of the decaying organic material in water. They're marine. They can be marine. They can be freshwater. They can also be present in soil. They're really very ubiquitous. But they have also been found on and within the tissue of different marine invertebrates. There are a few reports in the literature of them being found, again, within the tissue, actually, and also certainly on the surface of the animals. And other culture systems, like with tunicates, have also reported that the culture system supports the growth of these organisms. However, not everybody dislikes thrustochitrids because they actually are also seem to be known for their potential biotechnological applications because they produce a lot of polyunsaturated fatty acids, important ones such as icosapentaenoic acid, docahexanoic acid, but these are, very important polyunsaturated fatty acids that certain biotechnological areas have interest in taking advantage of their production of these for things and certain things. But anyway, I just wanted to show you a couple more pictures of these on-stain cells. If you look at some of the references in the literature, you can see that the ones that I showed, they're on-stain, those look very similar to the thrustochitrids that you collect in the water, unfortunately, or in the soil or something like that. And I cryopreserved them, and they come back out of the cryopreservation perfectly. So they're, it's really interesting, I guess, but not what I wanted. I did some other kinds of staining to try to characterize them. On the right, I did like an oil red O stain. So many of you are probably familiar that that's for staining lipids. And you can see that the lipid droplets, they stained red with this oil red staining, showing that these cells were actually producing quite a bit of lipid. So I did that. And then I also, on the left, you can see it's a cell flow cytometry histogram. From the literature, I found a fluorescent stain that researchers who focus on thrombocytosis use to identify thrombocytosis. It's a fluorescent stain, acrylflavine. So I use this for my cell cultures to try to use it as a way to identify contamination in my cell cultures. And this is just an example. On here, you can see the blue line is the day one of shrimp, when they were still shrimp. Well, it says no stain, but it was stained with a acrylflavine. I'm sorry, that's my mistake. And then the black was a day 91. So that's a later culture. That one has no stain. So the shrimp day one with acrylflavine and the day 91 with no stain are basically in the same spot. But the red line is showing the ones that are staining with acrylflavine. So about 50% of this culture was staining positive with the acrylflavine, in providing me more indication that that was a thrombocytrid. So that's just another method that we were using to confirm what our cell cultures were. We also did some different other staining with ray stain. Again, it's very variable. So I did get some cells that were nice big blue cells and blue nucleus with a very thin cytoplasm. And then, for another culture, you can get these ones that are mostly red staining. And then they have like occasional of the blue ones with the thin cytoplasm. And then you get these ones on the far right, which have a really large purple nucleus and a gray cytoplasm. So there was a lot going on in these cultures. And also they did stain. I did an alkaline phosphatase stain, and they did stain positive with the alkaline phosphatase. So that's basically where we got with the shrimp. We weren't able to get any further than that with the shrimp because we have, ended up with some challenges with getting additional fertilized shrimp eggs. I mean, we're here in Southwest Florida, and we did, our area got hit by, back-to-back hurricanes, and it did quite a bit of damage to facilities around the area, including the shrimp farm. So we have had challenges getting additional fertilized shrimp eggs. And, I think, moving forward, we would want to try different antibiotic type protocols to discourage thrombocytrid growth. And again, I wish I had focused more on those loosely adherent cells than I did, but again, I was focusing on the ones that were growing. We do have plans for doing spent media analysis and egg content analysis so we can understand what's in the eggs, and then understand what media is being utilized by the cells. I mean, what components in the media is being used by the cells. But basically, there is a lot of challenge with marine invertebrate cell culture that still need to be addressed by, many, in many situations. So that's what I'm going to talk about with the shrimp, and I'm actually going to switch the gears now to our redfish projects. We have a large aquaculture facility. It's not it's a site separate like from where the main lab is, but we have an aquaculture research park, and we raise several different fish species in an aquaculture setting. You can see the image of the farm there. And in the middle is a tank with red drum in it. So you can see we grow these fish in a big tank. And then they spawn, they're we can induce them to spawn, or they spawn naturally, but they're like when we get the eggs, again, it's from multiple fish spawning at the same time. So it's eggs from a group of fish that are spawning at the same time. So we collect them from our aquaculture facility that we raise here. And this is actually really ideal because we can get the eggs at exactly the right time that we want to after they've spawned, even when we have to wait for the fish to spawn. But once they spawn, we can make sure that we're getting the eggs at the right time. We can, check their viability and check the stage and things like that to get the eggs at exactly where we want them. So that's works out really well. Again, here is the red drum that we are using. They have that distinctive spot on their tail for identification. And here's an image of the fertilized eggs. You can see that they have like a lipid droplet in the center, and then off to the right or to the side, you can see the cell mass that's starting to form. And we basically collect the fertilized eggs about 2 hours after they spawn. In this image, you can see that PVC piping that we used a little easier. It has the nylon mesh on the bottom, and you can see the eggs are collected on the bottom. They're clear, and then they're small at the bottom of that we use for concentrating them and then releasing the cells. So red drum are also very important for the seafood industry. First of all, their name comes from, because they're named for the drumming or the croaking sound that they make with their swim bladder and abdominal muscles. People who live on the canals around here can really hear them at night, and sometimes it can be pretty disruptive, but that's where they get their name. Commercial fishing of the species is prohibited in Florida, so you can't, fish them commercially for sale. But they are a very popular recreation sport and a food fish. They do have slot restrictions and bag limits to, control the amount of fish that are fished. But, and then at our, excuse me, they are also raised through aquaculture efforts, raised for human consumption, and then, through aquaculture. So you can raise them in aquaculture and sell them in local restaurants and in some groceries. So, that's red drum. Again, I have a paper that summarizes this research, came out in, I guess it was late 2024, current research in food science about the initial efforts that we did with the red drum cell cultures. And in here, this slide, we can see that, I just wanted to show you how we collect the eggs. So we collect the eggs basically in a beaker, and this is like an egg sorter. And once they in the middle, so you can see the eggs in there, and we can get them out of there, through a little funnel, and it helps concentrate them and put them in this funnel, and then we can count the total number of eggs and discard all the unfertilized eggs, so we can determine which ones are fertilized and make sure we have fertilized ones, and then we rinse them a bunch of times and get the total viable eggs. But we used a similar protocol as we did for the shrimp for isolating the eggs. So first we collected the eggs, and then you can see the sieve there. We took that sieve and we put it through a number of washes, PBS, a 70% ethanol, another PBS, a brief bleach, a dilute bleach solution. And again, we had PBS++, which had the penicillin, streptomycin, and the amphotericin to help microbial growth from the outside of the eggs. We crush them, like, again, with the same style of borosilicate vial to release the eggs into the petri dish below, remove the debris with like a centrifuge top filter. And you can see a small picture there of the isolated cells from that. And I'll show you those in a minute. But again, this is the red drum eggs after spawning, about 2 hours. You can see the lipid droplet and the cell mass. For most of our so we did test other medias, but the one that actually has been most successful for the red drum cell culture to date has been using a DMEM, a high glucose DMEM from ATCC, which we add fetal bovine serum to, pan-strep, amphotericin B, a herpes buffer, leukemia inhibitory factor, fibroblast growth factor, sodium selenite, sodium pyruvate, and the osmolarity of this is just about 300 milliosmolar. But that's what we have had the most success with. Oops. Okay, so here are the cells at day 0 up at the top. You have large round cells that, they're not completely uniform in size or look, but they are, a lot of the big, larger round ones. And down below, I did a rice stain on day 2, and you can see that you're starting to get some types of, eosinophilic staining with the rice stain too. So giving us some indication that maybe it's more hematopoietic development or differentiation into hematopoietic type cells. So we did a Western blot with our the lysate. We did a lysate of the red drum cells isolation time to try to understand if there were any types of transcription factors that were found in stem cells of other organisms. And to be honest, we just used commercially available antibodies that most of the target species were like human or a mouse, but we just tested them with our red drum lysates. And the blot that you see here is with OCT4, the octamer binding transcription factor 4. It's very often used in different to identify different stem cells in different species, and we had a good response to that. But we also tested a panel of other ones. and will be in that paper, but they are stage-specific embryonic antigen, NANOG, SOX2, alkaline phosphatase, and proliferating cell nuclear antigen. And these were all positive in the red drum lysate. So, we were happy that that was giving us some indication that these are maybe some, early stage cells for our cell cultures. Okay, so this is what the first attempts looked like. Day 0, I showed that already. But day 1, you can see that they're starting to get some adherent cells. That's an untreated plate, and you can see some cells that are starting to spread out on the flask, and you have the round ones kind of floating in the suspension. And then up in the top right, you see a day 2. This is on a fibronectin treated plate. You can see the cells are starting to spread more, but they're not very concentrated at this stage, but they're starting to grow. And then day 7 is down below. That's an untreated plate. Again, we tested some fibronectin and untreated, but we ended up just sticking with it untreated because it didn't seem to make a huge difference. But as of right now, we do have red drum cells that are in culture for, I did this last week, but for over 232 days and over 73 passages. And right now we have like 2 that morphologically, so this is only a morphological identification that seem to be like fibroblast or more mesenchymal. And we've been able to determine their doubling time, and this is decreased over time. So early on, the doubling time was around 60 some hours. Once they got to a stage where they were, in a high enough concentration that we could determine the doubling time. And then, through more frequent changes of cell media, I think kind of selecting for ones that are dividing more rapidly, we've been able to get the doubling time down to 24 to 35 hours. And this is just showing some of the cultures of those. On the left column, you can see the ones that I are calling to have more of a fibroblast morphology. That's day 158. So these are longer term cultures. And day 234, they're stable. So I just put those 2 on there so you can see that the morphology hasn't changed over that time. And then in the other pictures, you can see these, it's probably really hard to see, but these cells, because they're unstained in that first picture, the day 158, but the cells have a lot of vacuoles in them and they're adherent and they form like a really solid mat on the bottom of that flask, but they have a nucleus and then those vacuoles. I tried to stain it with a hematoxylin to the right of that, and you can see the nucleus and the vacuoles a little bit better. And down below is a continuation of the culture day 234. You can see kind of this cobblestone. It's their cobblestone appearance under the microscope. And again, if you look able to see it more closely, you can see that they have a lot of vacuoles in them. This is with a right stain. The one on the left is the ones that look more fibroblast, and then the ones on the right are the ones that are more mesenchymal. You can see more clearly the purple nucleus and the gray cytoplasm, and then the different vacuoles in the cytoplasm that I was referring to. Okay, so more recently we've been able to take these cells and grow them on different, we're trying to grow them on different scaffolding. These are ones that we obtained from Cytonest. And these are day 45 culture. So, and this is a polycaprolactone scaffolding, and then also one that was treated with plasma. And you get, the reason I'm showing day 45 is because the earlier days you didn't have a lot of cell culture. So it took a while for them to adapt to this to this scaffolding, but they have adapted to it now. And then you can see that they're starting to fill in pretty solidly. I mean, especially with the distance between the scaffold fibers is less. They fill in, seem to fill in a lot easier. And then the one on the right is with the plasma treated, and they seem to prefer the plasma treated as well, as far as forming more of a solid cell sheet on that scaffolding. We also did experiments where we tried that same scaffolding with a matrigel. And this is an earlier time point, so this is day 11, but they did seem to grow better in the matrigel because they filled the scaffolding space a little bit more quickly than it did when without that. So these are just some experiments, qualitative experiments that we did to see which surfaces they were growing better on. All right, so that's really where we are with the red drum right now. Our research is ongoing. Of course, we want to attain more shrimp cells, but we have a lot to do with the characterization still of these red drum cells because we are really excited about how well they're growing in a culture. But we need to focus on characterizing them and also some differentiation efforts to try to differentiate them into muscle or adipose cells because right now they're growing very happily in what I'm giving them as what they are, but our goal is to differentiate them into something that's more relevant for cellular agriculture. Again, we also have plans for their egg content analysis and spent media analysis for these, and also some transcriptomic studies that we're planning to as well to understand what's happening in the cells with different media types. And, they're growing pretty well in the scaffolding, but from what I understand, is way too slow to be useful. So we're, working on trying to improve different ways to get them to grow more quickly and, and more into what we, more type of myoblast type cell line or something like that. So we have a lot of work ahead of us, but we're really happy with the way those cells are growing right now. So I just want to provide some acknowledgements. Of course, Amy had mentioned in the beginning some funding from the Good Food Institute, current funding from USDA, and also our collaborators out at the Aquaculture Park. So we appreciate everybody's help with this project. And that's it. So I have, if anybody has any questions, I'm here to answer them. Okay.
Thank you so much, Dr. Walsh. That was really fantastic. And we do have a number of questions flooding our Q&A section of the Zoom bar. So first of all, thanks again, Dr. Walsh,
You're
for a
welcome.
fantastic seminar. There's a lot of folks in the chat who kind of relate to this persisting problem of contaminations in seafood culture. So maybe we'll start with a first question around how, despite extensive sterilization procedures, many often experience persistent contaminations. This person's work with shrimp was plagued by repeated bacterial and parasitic colonization. And it's just kind of curious if you could kind of summarize your perspective on best practices for dealing with this. That might be really helpful for the community.
Right. Yeah. So, I mean, obviously, I don't have the answers. Otherwise, I wouldn't have that problem. But I mean, honestly, because I feel like we really did our best efforts that we could to reduce contamination. So I think understanding how to control the bacterial contamination, having everything be sterile from the beginning to the end, and you have to include antibacterial agents in your cell culture media. I know that that's not going to be ideal long term for cellular agriculture, but I think to get some of these cultures started, you're going to have to work with that. And shrimp, that's really difficult because they do have a lot of susceptibility, even in aquaculture settings to different viruses and stuff like that. So that's a big problem in the shrimp industry, to start out with. So I think honestly, and also I'm not sure where that person who asked that question, where they're getting their cells from. Is it from like muscle or hemolymph or where it's being obtained from? Because that, can also impact. So I mean, you obviously, if you're getting it from like the shrimp itself, like drawing out hemolymph or something like that, making sure that the surface is aseptic too, putting them in some kind of cleaning off the surface really well, and then just trying a different battery of types of different antibiotics to try to keep it minimal. But yes, it's very, very much frustrating. Now, to be honest, we really didn't have with these eggs that the way we were getting them, we didn't have trouble with bacteria contamination. We just had the throstochytrid. So that was not a big issue. And I don't know if it's because the eggs were the way we were getting, we weren't having to go through the animal, like even to get the muscle out, you have to, open up the animal and get into the organism itself. And I think that they just have a lot of susceptibility to different microorganisms, their tissue do.
Totally. Sounds like a persisting challenge that we'll need to
Yeah.
continue to better understand and adapt to. There's a question here about the challenges you experienced around the Thastra chytrid population. So just a clarification point, did you find that the Thastra chytrid population completely outcompeted the shrimp cells for the nutrients in the media? Or did the shrimp cells begin to die and then the Thastra chytrids began to proliferate on the decaying cell matter?
Well, honestly, probably both. I mean, I think it was honestly probably both. I think definitely the thrusted chytrids outcompeted the shrimp because they were loving whatever I was giving them. But I think that as the shrimp were dying, I'm sure that they were eating them up because there wasn't like dead shrimp cells lying around. It was like a very clean culture. So I wasn't like, you know, uh, uh, thinking that, wow, all these cells are dead in here. So I think that they did. I think it was both.
Super interesting. There's a question here about culturing embryonic cells of freshwater neotropical fish. So this person is just curious whether you have recommendations for culturing embryonic cells of freshwater neotropical fish that grow in low osmolarity in particular. Is it possible to use the same medium as is used for insects? This person's work with the species has run into a lot of challenges keeping cells alive in culture.
Uh, Well, I mean, I don't know what you mean by low osmolarity. I mean, for typical mammalian systems, it's around 300 milliosmolar. Most of like the commercially available cell culture media will be around that um, as well. But I think you need to prepare your media and test what the osmolarity is, so it can be more appropriate for whatever the level that you're trying to get. But yeah, I think you could. I think you just need to adjust whatever you're using. And sometimes what I've done, I haven't ever gone down in osmolarity below 300 milliosmolar. I've always gone up because I also work with like a lazarbrite species and they have like 1,000 milliosmolar. So sometimes what I've done is gotten like the powdered form of the media because then you can adjust it a little bit better, as far as having to add a lot more like salt or urea or things like that, like that I'd added for lazarbrite systems gives you a little bit more flexibility to adjust the volume how you want. But I haven't really gone down. I mean, you just have to adjust it to what it needs or do some tests because sometimes they have a range, like they can be um um so is the egg content, I guess one of the questions I have is the egg content itself low in osmolarity, or is the physiology of the fish, like the physiological fluids, lower in osmolarity? Because sometimes they do have a range, like even though they're at a certain osmolarity, they're more tolerant if you can go up a little bit or something like that. So you could do a test of the different osmolarities and see, you can look at them under the microscope to see if they're, in the appropriate osmolarity or not, because they'll shrink or swell. But yeah.
Awesome. Thanks for that advice. Hopefully that's helpful, Jennifer. And sounds like Dr. Walsh is welcoming some cross dialogue. Um, so there's also a question here around what the importance and significance is of supplementing the culture medium of seawater fish with sodium selenite. Can you speak to that a bit?
Um, honestly, I put that in there because I was getting that off of another medium that was published, and so they had sodium cianide in there. But it helps as an antioxidant in the cell cultures. Now, if you put too much in there, it can kill them because I did do that too. So you have to be careful of the level. So that's really the rationale behind that. It was in another paper that I was, looking for advice, from everything that I could find, and they had use it to put it in there to help be an antioxidant, basically, to help maintain the oxidative state of the cultures.
Thank you. There's another question here. You talked a bit about foreign cell contamination and its challenges that need to be eliminated. And this person is wondering whether you see potential for invertebrate culture systems to embrace a monoxenic or polyxenic culture system by adding other species selectively and then intentionally co-culturing them with a target cell type to support survival and development.
Well, yeah, I mean, and I haven't tried that, but I think it's worth a try because I'm not sure the culturing them individually is gonna take a lot more. it's going
Mhm.
to take a work because I feel like a lot of people are running into challenges with trying to culture, like for example, the shrimp cells individually. So yeah, a cold culture system might actually be more successful. I haven't really tried that, so I would think it's definitely worth a try because I feel like a lot of us are kind of going down the same road and running into similar problems.
Mhm. Absolutely. Awesome. Well, that brings us to the end of our audience questions. There's a whole lot of gratitude in the chat and throughout. Just thanking you for taking the time to talk about your work today. It's such important foundational work for the cultivated seafood theory of change, and really is all of this cell culture work is the first gate that we have to get through if we're to even have a fair chance at sustainably and scalably feeding the world through these novel food technologies. So, very much appreciate this really foundational work. And thanks to all of you for being here today. I love to wrap by giving our speakers a chance just to say any last words with the scientific community that's here. So, if there's anything you'd like to share, with all these scientists who are calling in from around the globe, feel free to. We'd love to hear any advice you have.
Well, I don't know. I think I was trying to give my advice throughout, so I'm not sure everything else to add, but I really appreciate everybody's attention today. And, feel free to reach out to me. I did put my email on that first slide. So if you want to, reach out to me, I can, you know, if you have any questions, I can, try to answer them as best I can. But, obviously, as you are aware, there's just so many things that people haven't tried yet or don't know what would happen. And,
Totally.
it's a lot to do still in this field. So I think it's pretty wide open. So it's exciting in that way, but there's some really big, challenges like, the contamination is just something that we need to get past, but and I think that we can be successful. I just gonna take some more work for sure. So as a community.
Absolutely.
Mhm.
Yeah, a wide open canvas and like really ripe for collaboration. I think the cultivated seafood
Mhm.
community in particular is really tight knit and interconnected and excited to welcome new folks into the fold. So thank you all so much. Thank you, Dr. Walsh, for your work.
You're welcome.
So incredibly important. And look forward to seeing everyone again next month for our monthly Science of Alt Protein seminar. Thanks so much. Bye.
Thank you. Have a good day.
You too.